Great. I think we have about as many people here by Zoom today as we do in person. So I do appreciate those of you that come in person. I think there is some value to that. I know there's times where that might not work out, but I think it is good to get out and see people and try to talk to people, and you have the chance to ask questions. Not very many of you have, but please feel free to do that. So looking ahead, next Wednesday is our next exam. That's going to cover the antibiotics, the process of infection, the innate immunity that we finished last time, and the adaptive immunity that we'll finish this week, sometime on Wednesday. I'm still not sure we'll wait and see about the vaccine stuff. Right now I have the chapter 17 vaccine material. That homework is set to be due next Wednesday, but there's a chance that we'll push that off. I don't want to rush the vaccines this semester. Okay, so just kind of a heads up with that. I'll try to make some announcements about that next time. And I thought we would just start with a few review questions. It's been a while since we've done that. The antibiotic stuff is a little bit more just kind of, you gotta memorize it probably. The chapter 14 infection stuff is a lot of vocabulary, really. So those things are a little more cut and dry. We get to the immunology stuff, it's more processes and sometimes a little bit harder to wrap your head around. All right, so here's an example of a question used on a previous exam. Which of the following is a semi-synthetic drug that blocks the action of RNA polymerase and is often used to treat tuberculosis? All right, so correct answer here should be rifampin. That was the one that also stained bodily fluids orange because it was an orange pill. So that might have been another thing that I could have said to give you a hint. Remember, for all those antibiotics, I think we had about 12, 13, 14 of them that we went through. Know especially their mechanism of action. So here I said, blocks the action of RNA polymerase. I gave you a lot of other information, like maybe how it was administered and what kind of diseases it might be used for. I try not to, I want you to especially know that mechanism of action. I might give you some other information, you know, that would be helpful. We talked about knowing advantages, disadvantages, you know, drawbacks, but mostly the administration and, you know, like whether it's a natural or semi-synthetic, I didn't even necessarily mention that, but I wanted you to get familiar with using those terms, like what's the spectrum? Is it narrow spectrum or broad spectrum? You know, is it susceptible to degradation by stomach acid? You know, that means acid labile. So I wanted you to get kind of familiar with those terms and not necessarily have to know every little thing about all those antibiotics. All right, I'll give you a second to read this one and think about what your answer would be. Under which of the following conditions will penicillin most likely be ineffective? So in other words, when would penicillin not work? If you're online, make sure you're muted. Thanks. All right, so let's go through these. So when would penicillin not work? If the body temperature was higher than normal. That we talked about the effects of fever, but none of those are going to be high enough to make a drug not be working anymore. So that's not right. If the concentration of the drug is too high, I don't think that's a problem. It could potentially be too low, but I don't think too high is a problem. If penicillin is administered intravenously. So the first penicillin that we came out with had to be administered by either IV or injection in order to work. I don't know of any drug, well, there might be a few examples to that, There's some that we don't administer that way because of toxicity issues, but it wouldn't, in penicillin's case, that drug always works when we, again, give it by IV or injection. It may not work if we give it orally. All right, so that could have been a little bit confusing. We talked about therapeutic index. That doesn't really have any effect on whether the drug works or not. It might, again, make us not want to use it. If the therapeutic index was too low, we wouldn't want to use it because it's too toxic to the host. But again, that's not going to have any effect on whether the drug will work or not. So if the target cells aren't actively growing, then penicillin won't be effective, because remember, the penicillin mechanism of action interferes with the cell wall synthesis. Okay, it interferes with those transpeptidases that are sealing up those bonds in peptidoglycan, and if that's not able to do that, the cell wall just won't be intact, and the cell is more susceptible to bursting by trigger pressure. It was a long explanation, maybe longer than it needed to be. Okay, one more. This is from chapter 14. I'll give you a second to think about this one. Endotoxins and exotoxins, it's easy to confuse those. Which of the following types of bacterial toxins can be produced by both gram-positive and gram-negative bacteria? Right. Well, if you remember what endotoxin is, remember endotoxin, that's the same thing as lipid A, which is found in lipopolysaccharide. And remember, that's found in the outer membrane of gram-negative bacteria. All right, so endotoxin is only in gram-negatives. All right, so the correct answer here would be A. Exotoxin is potentially made by both gram-positives and gram-negatives. All right, makes sense? All right, so where we ended last time, we were just finishing up chapter 15 with the innate immunity. We kind of, at the end, rushed through the interferon, how interferons work. I also had posted this just picture of this table from your book. It's a nice sum up of our innate immunity. So it splits it into the first line and the second line of defense. Remember, those are artificial constructs like that we've put on in order to help us kind of organize how we think about innate immunity. I'm not so sure that it's all that important to, oh yes, definitely the cells are in the second line. Typically, once the first line is breached, we think about the first line as being protective barriers like our skin, maybe our normal microbiota is kind of an intrinsic barrier. I noticed just today that this table in the 6th version, the most recent version, is a little bit different. It considers, I think, at least phagocytes, extracellular killing, and maybe even complement as the first line. But then in the text, it talks about as being second line. So I would go, let's go with this one. Again, it's not a point that I'm going to get hung up on, but it's a nice little way to review innate immunity. So again, we talked about barriers. We mentioned a few specific chemicals in there, like lysozyme. Remember that breaks up those glycosidic linkages between the sugars and peptidoglycan. Not too much specific there in terms of associated chemicals there. We talked about cells in the second line. So ones that are phagocytic, macrophages, neutrophils. We talked about cells that are granulocytes or agranulocytes. So try to keep all those kind of straight in your mind. Monocytes, they differentiate into macrophages and dendritic cells, like what all those cells do, how they will act on pathogens, different ways that they do that, as well as, maybe degranulate or phagocytize, for instance. Complement proteins, so Main idea there, there's a group of proteins that can complement other aspects of immunity. Bits of them can act as opsonins, which, remember, an opsonin is something that stimulates phagocytosis, can increase inflammation. It can cause these membrane attack complexes, which are especially useful against gram-negative bacteria. We ended last time thinking about a particular cytokine called interferons. They're especially useful in fighting viral infections. It's, again, we'll just briefly overview it there. I am not actually sure what it means specifically about antimicrobial peptides. It may just mean cytokines in general. It would be nice if it would list something a little bit more specific there. The action of inflammation. So blood vessels swell up a little bit. The spaces in between the cells get a little bit leaky. And that's the mechanism where cells can leave the blood vessel by margination and diapedesis and to go into the tissues to fight infection. And lastly, we talked about fever, maybe minimal help in terms of fighting an infection. We talked about how usually just a degree or two increase isn't going to be enough to really slow down the replication of some pathogen. There is one notable example that I wanted to mention last time, and I totally forgot. There's a bacterial infection called syphilis, which we don't hear about anymore because we have had antibiotics that take care of it very easily. But it was a really prevalent sexually transmitted disease, 100 years ago. Caused, lesions in places you don't want lesions, rash, and eventually there could be long-term consequences like dementia and heart damage. That this syphilis could kill you. And it was discovered, though, that bacterium was really sensitive to heat. So somebody realized he was his name was a name that we've forgotten now because his treatment's not used anymore, but his name was Julius Wagner Jauregg. And he got the Nobel Prize in 1927 for realizing that he could infect syphilis patients with an infection that caused a high fever, malaria. Okay, and then that high fever caused by, in a response to the malaria pathogen, would kill the bacteria from syphilis. So again, cure for syphilis, it was great. We must have had a little better treatments for malaria at the time. Otherwise, pick your poison. What do you want to die from? But again, once antibiotics came out, that treatment became obsolete. Okay. So just a little review there. So where are we going today? We have been through, again, the first and second line. I don't think I posted this again because I posted it previously. We're going to focus now. We're moving into chapter 16, adaptive immunity, where we're going to focus on our specific response and mostly focusing on B cells and T cells in the role that they play. I would encourage you, if you haven't read your book very much so far, to start reading a little bit in chapter 16, especially as we get into next lecture where we're talking about B cell activation and T cell activation. That's when it starts getting kind of complicated. So read a little bit about that. And you're going to be better prepared for Wednesday. Once we get through adaptive immunity, our material gets much more straightforward. We're talking about specific infections, pathogens, and it's generally a little easier material for students. So get through this week, and then, sort of the end is in sight. So I know the semester fatigue is starting to set in, but try to power through a little bit longer here. All right, so adaptive immunity. So we're going to start with just kind of a little overview how adaptive immunity is different than innate immunity. And then some a little anatomy. So what are we talking about? We're talking about lymph and lymphatic system. What kind of structures are we talking about in our body? I've tried not to use this word too much so far, antigens, because I haven't really defined it yet. So we're going to define what that means to be an antigen and what molecules make good antigens. And then antibodies are what we make in response to antigens. And we're going to look at their structure, how they function. Then we're getting more into T cell function and think about how T cells work, how cells advertise that they are infected by a virus. Okay, that's cytotoxic T cells that act on virally infected cells. And just kind of at the end there of this chapter, thinking about T cells and B cells become activated. That's a process that has many steps to it, and sort of, we want it that way because we don't want to have inappropriate immune responses. We want to make sure that it's warranted before our immune system starts firing. So, all right, so a little overview of adaptive immunity. So the body's ability to recognize and defend itself against distinct invaders. All right, so 5 attributes of adaptive immunity. Number 1, specificity. So responses are tailor-made. That's a little bit misleading the way I define that, because we actually are not making antibodies once we're exposed to some pathogen. We actually already have all those antibodies kind of ready to make, and then we just kind of activate them and make more and more of them. So, but they are extremely specific, and hopefully you'll understand that today by looking at some of the structures here that I've got for you today. Let's see, inducibility. Okay, so like I said, we already have these T cell receptors and B cell receptors that recognize almost anything, any potential pathogen that we could come in contact with, but only a few of them. So they have to be activated when they're needed and make a whole bunch more of them. They have to proliferate. So we call that clonality. Okay, make lots and lots more of them. Unresponsive to self. I thought that was fairly self-explanatory there. But we have mechanisms in place so that our cells are not harmed by our own immune system. And of course, when those mechanisms fail, then we get autoimmune disease. And lastly, a hallmark of adaptive immunity is memory. Okay, so the next time that pathogen is encountered, we have a much faster, more vigorous immune response than the first time because we have some memory cells. All right, so that's a big difference from our innate immunity. So there is actually, we think now, so conventional wisdom, right, innate immunity doesn't remember. You get the same response every time, where adaptive immunity, we get a little different response, we learn. There is actually some learning that can take place with certain kinds of innate reactions, but I guess, you know, everything's more complicated than what an introductory course would make it seem. So again, there is a little bit of learning that goes on there, but nothing like what we see with B and T cells. All right, so like I said, adaptive immunity involves the activity of lymphocytes. And so apparently when we stain B cells and T cells, they're almost impossible to tell the difference between them just by staining them. So we have to do, I don't even know how you tell the difference. You have to look at their receptors perhaps to tell the difference between them. So B cells, they're going to be responsible for our antibody immune response. Sometimes we refer to that as our humoral immunity. Your body's humors are the fluids, okay? So antibodies are in the bloodstream, so that's why it's called humoral immunity. Okay, these antibodies are going to be best at acting on extracellular pathogens. Okay, so as well as toxins, I'll add that. Okay, so invaders that we would encounter outside of cells. Okay, so again, that could be a toxin. Viruses for a time are outside of cells, and then they're going to move into cells. Harder to find them when they're inside our cells. T cells are responsible for cell-mediated immunity. And they are our mechanism to act on intracellular parasites or pathogens, I mean. Okay, so an intracellular pathogen, all viruses are going to be eventually in our cell because they can't replicate unless they're inside of a cell. Some bacteria need to be inside a cell in order to replicate as well. There's only a few examples of that, but there are a few. Okay, so you could sort of think about our B cell, our antibody immunity is kind of like, in terms of defense, as bullets maybe, kind of against some enemy, where our T cells are going to act more like hand grenades. They're kind of our heavy duty machinery. Maybe a tank would be a better analogy, because the T cells are actually acting to destroy our own cells that are virally infected. And antibodies are mostly just sort of helping out. All right, so before we talk more about B cells and antibody, we need to talk a little bit about where these immune reactions are going to be happening. And that's in the lymphatic system. So this is where we find lymphocytes. Okay, it makes sense in the lymphatic system, traveling in the lymph. And you could kind of think about lymph as being similar to blood plasma. All right, so it's a lot of fluid, it's some cells, it's some other proteins and things in there. But the lymph is traveling through our lymphatic vessels. It's moving in one direction. We'll see a kind of a close-up of a lymphatic capillary in a minute. But it's moving in one direction, and it's going to end up here kind of near our heart, in a place where the blood pressure in the body is the lowest. Okay, and that's in the subclavian vein. So in the subclavian vein, the lymph actually dumps back into the subclavian vein. Okay, so lymph goes in one direction and dumps in there. And then where does the lymph come from? It comes out of the bloodstream and enters the lymph. So we'll look at that in a minute with a couple different pictures. All right, so primary lymphoid organs where lymphocytes are made. So conventional wisdom is that lymphocytes are made in the bone marrow. And I say conventional wisdom because I, until last week, thought that's where all lymphocytes were made. I was doing a crossword puzzle, and that was the clue, where lymphocytes are made. I thought, oh yes, one that I know, but it was only 6 letters. And it turned out the correct answer was spleen. I'm like, that's not right. Well, I went and looked it up, and actually some lymphocytes can be made in the spleen as well, but most of them are made in the bone marrow. And not just any bone marrow, but it's going to be, red bone marrow. So flat, bigger bones like your femur, I think in your pelvis. Okay, so that's considered one of our primary lymphoid organs. B and T cells are produced here, mainly here. And B cells are going to mature here. Oops. So what does that mean, mature? Basically, it means that they get their T cell receptors and make sure, you know, they have B cell receptors that are going to be functioning and not targeting the wrong things. All right, so get the B cell receptors. Another primary lymphoid organ is the thymus. And lymphocytes aren't made there, but this is where T cells mature. Okay, so they travel from the red bone marrow into the thymus. The thymus is a little gland near our heart. So in the thymus is where T cells are going to differentiate into different kinds of T cells. And again, they get their T cell receptors and make sure that those receptors are not targeting something in our own body. Okay, we want them to only recognize pathogens. Right, and most of that work of maturation, at least with T cells, happens before birth or just after birth, surprisingly enough. So as we age, that thymus gland kind of shrinks more and more and doesn't really do much of anything as we enter adulthood, which is kind of surprising. Okay, and then secondary lymphoid organs, this is where lymphocytes are activated. So this is where B and T cells have a chance to mingle with other kinds of cells like macrophages and dendritic cells that would present antigen to them or present bits of pathogen to them. All right, we have somewhere between 500 and 700 lymph nodes. These are about the size of a pea. So we got a ton of them all over our body. They're especially concentrated in our neck, armpits, and groin. And again, this is where in the lymph nodes, especially that the cells that are B and T cells have a chance to kind of mingle and match up with their receptors to certain bits of pathogens. Okay, and the lymph nodes have kind of a tough outer coating to them. The spleen, here's the spleen over here. I already mentioned that its main function is to filter the blood. So where the lymph nodes filter the lymph, the spleen is going to filter the blood. When I say filter, we're especially good at recognizing, you know, clumps of pathogens. So we have mechanisms like antibodies that helps pathogen clump up. Okay, that gets filtered out in the spleen. You can lose your spleen and live pretty normally. You're going to be a little bit more susceptible to certain, especially bacterial pathogens. You might need to really be diligent about keeping up on your vaccine schedule. You might need to take antibodies prophylactically if you encounter somebody that, you know, exposed to somebody that has some certain infections. But if that spleen gets ruptured, maybe as, you know, an accident of some kind, apparently it can't just be sewn up. They have to take it out. Okay. Tonsils. We can also do without our tonsils. Some of us are better off without them. They actually function just as another set of lymph nodes. So we can do without some of our lymph nodes and be just fine. Some people, they're particularly large and, you know, bacteria get kind of caught in the crevices of it and have problems with repeated infections, and we're just better off without them. MALT, that might be a little bit not as familiar. That stands for mucosal associated lymphoid tissue. And this is tissue that's basically functions like a lymph node, but it doesn't have kind of the hard outer coating of it. And we have mucosal associated lymphoid tissue associated with all our mucosal membranes. So our respiratory tract, our digestive tract. We have a special name for some of them that line the intestinal wall, the MALTs that lines the intestinal wall and kind of samples inside the small intestine looking for foodborne pathogens. Those are called Peyer's patches. Okay. Let's see, what else did I want to say? All right. Okay, questions about lymphatic system? If we look at a close-up of the lymphatic capillary, how it's in close association with a blood capillary. Okay, then you start to understand kind of where the lymph comes from. So, you know, we have blood, it's, you know, being pumped right through our blood vessels from the heart here. Normally we have things that are secreted, you know, leaking out or migrating out of the blood vessel wall. Of course, we've got an exchange of gases, we got oxygen moving out there, maybe have a little bit of water leaking out, especially during inflammation, but maybe other times as well. You're going to have some cells leaving the bloodstream. And other things like nutrients, maybe some proteins, things like that. Of course, the cells are leaving by margination. They're going to slow down and adhere to the cell wall. And then diapedesis, that's the process by which they leave the blood vessel. Okay, and anytime the pressure in this kind of intracellular fluid here is higher than inside the lymphatic vessel, these things will migrate into the lymphatic capillary. All right, so fluids, you know, especially after inflammation, we've got, you know, more fluids leaving the blood vessels. Okay, those fluids end up in the lymphatic capillary. There are, of course, we don't have a lymph pump. All right, so how are things moving through our lymph? Smooth muscle contractions, to some extent, maybe some skeletal muscles as well. So it's always good to kind of move around and get your lymph moving a little bit, maybe. But there are valves in here so that the lymph can only move in one direction. Okay, and again, it's going to be traveling towards the heart, that subclavian vein. That's where it's eventually going to dump back into the bloodstream. So lymph is, again, a lot kind of similar. You can say it's similar to the plasma in our blood. Okay, red blood cells aren't ever supposed to leave the bloodstream. So other kinds of cells could be, though. Okay. And then if you look at a cross section of a lymph node, let's write this up here. You'll see the lymphatic vessels coming towards the lymph node. Those are referred to as afferent lymphatic vessels. And that A means towards, so towards the lymph node. You can see there's 1, 2, 3, 4 afferent lymphatic vessels in this picture, and only 2 efferent ones. Efferent meaning away. Right? So anytime you've got, say, more of something, say more people coming into a room and only maybe one exit, that's going to create a bottleneck. And that's by design, because you want those cells, say they're macrophages that are presenting, you know, a piece of some pathogen. This is where the macrophages are going to intermingle with the B cells and T cells that need to kind of match up their receptors. So we've got, again, a chance for cells to mingle a little bit there. And that's ideally what we want. So during an infection, it's really common for lymph nodes to swell up. Okay, that's not too surprising, because you're going to have, you know, during inflammation, you're going to have more fluids, you know, more lymph, more cells. Sometimes there's so many cells in there, white blood cells, that, you know, some of these, maybe the afferent vessel could get stopped up a little bit and the lymph nodes swell up. So that's one way we can tell if there's an infection going on. All right, so this term antigen, I've used it a couple times and tried not to. Some of you know what an antigen is already. I'll be more intentional about defining it and thinking about what makes a good antigen. Antigens, we'll call them molecules the body recognizes as foreign. Okay, So if you look at the word, these are actually called, we named them antigens because they're antibody generators. Okay, so antibody generators. So sometimes I've had students say to me, well, I get mixed up, what's the good thing and what's the bad thing? The antibodies are the good thing that we use to fight infection. The antigen is what causes the antibodies to be made. Okay, so what can function as an antigen? Really, when I talk about something functioning as an antigen, I mean, how well is it eliciting some kind of an immune response? Okay, so a good antigen maybe elicits a really strong immune response, and something that's kind of a weak antigen doesn't, you know, our body doesn't recognize very easily as being foreign. So what kind of things function as antigens? Various bacterial components, proteins of viruses, fungi, maybe even protozoa. Something doesn't have to be a pathogen to function as an antigen. If you have allergies, you know that. So food, you might have a shellfish allergy, for example. That's because your body recognizes something in shellfish as foreign and you have a reaction to it. Dust or feces and dust mites, okay, people recognize as foreign. So again, it can cause immune reaction. The parts of antigen that our immune system recognizes, we have a special name for that, and that's called the epitope. Okay, so regions of antigens that are recognized by our immune system. Okay, another name, I'll write it up here. Another name for this term epitope is antigenic determinant. Okay, I don't typically use that term. I typically use the term epitope. Okay, so for example here, if the bacterial cell is the antigen, okay, there's lots of different, you know, maybe sites or different molecules on the protein or the bacteria's surface that could act to elicit an immune response. So there's lots of different epitopes here that the, you know, different antibodies might recognize. Might recognize flagellin or, you know, whatever surface, anything on the surface there, lipopolysaccharide. Okay, so we'll get comfortable hopefully using those terms. What makes a good antigen? So in other words, what properties of antigens allow it to elicit a strong immune response? What's strong? Okay, really three things that we look for that makes antigens most likely to elicit a strong immune response. And these aren't hard and fast rules, they're general guidelines. But if we look at the shape of an antigen, anything that has a very distinct shape is going to be more likely, more easily recognized. All right, so maybe for a protein that's alpha helices or beta sheets, something very distinct about the shape. Large antigens, large molecules are much more easily noticed. Okay, and again, there's not hard and fast rules here. Generally, when molecular weight is below 500, it's just not big enough for our immune system to recognize. There is an exception though. If a very small antigen links up with some carrier molecule, and then is big enough to get noticed, we call that small antigen a hapten. Okay, so a hapten is a very small antigen, very low molecular weight that is able then to be noticed by our immune system. So an example of that, poison ivy oil, okay, is actually a really tiny, tiny molecule, and it shouldn't be big enough to be recognized by our immune system. but I had poison ivy 3 times 2 summers ago. So we know for a lot of us, it is. What's going on there? That small molecule links up with some serum proteins in our blood when it soaks into our skin and links up with these serum proteins, and then that makes it elicit more of an immune response. And lastly, complexity of a molecule. Okay, if it's varied, if a molecule is not just the same thing over and over again, then there's going to be a lot more epitopes for the B and T cells to recognize. Coronavirus, right? When we're talking about fighting coronavirus, all you hear about is these spike proteins. All the vaccines are targeting the spike proteins. So what is it about the spike proteins? Well, there's nothing else on the surface of that virus that is antigenic at all. Just these spike proteins. And so that's what all of our therapies basically are targeted towards. Okay, so if you think what kind of molecules fit those 3 things, shape, varied shape, the shape and complexity are a little bit kind of the same thing. Large size, high complexity. Well, protein molecules are generally what we think about as being the best kind of antigens. So for example here, this molecule here is beta galactosidase. And you don't have to know that. That's just an example of a protein. It's the protein that splits lactose into glucose and galactose. That's a very complex molecule. Most proteins are. They have different kinds of folding. They have all sorts of different functional groups on them, different amino acid sequences. So they're very complex. They're pretty large. They have unique shapes. They hit all the boxes there. Okay, so our immune system is generally going to be triggered by them. Molecules that are, like lipoproteins, like part lipid, part protein, or glycoproteins, part sugar, part protein, are kind of mid-range. All right, so yes, they are usually antigenic. Something like polysaccharides are usually very poor antigens. Again, that's not a hard and fast rule. There are exceptions to that. But here's an example of a polysaccharide, starch. Okay, glucose, 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 glucose. Okay, the same thing repeated over and over again. Not very complicated. Okay, it might be big. That's kind of the same thing. So that typically is again going to make kind of a poor antigen, but there are exceptions there. Okay, something like DNA is typically not very antigenic, but there are exceptions to that as well. So kind of looking at all our macromolecules. All right, a little further terminology to do with antigens. Antigens that have entered the body from the outside. Okay, we call those exogenous antigens. Okay, so, any kind of bacteria that comes into some portal of entry, whether you breathe it in, whether you ingest it, okay, it starts outside of the body, comes into your body. So that's an exogenous antigen. And then an endogenous antigen. It's inside of the cell or is produced there by some intracellular pathogen. So like a virus that is inside of the cell. So viruses start out as exogenous, when we breathe them in, for example, but then when they come into our cell, then we would consider them endogenous. Okay, so certain parts of our immune system are good at targeting viruses when they're outside of the cell, but then once they're inside of the cell, they don't work anymore, and different aspects of our immunity have to take over. An alloantigen is an antigen that's originated within the cell and only some members of the species have it. All right, so for instance, something that wouldn't be antigenic for me, but then if it was put in your body, it may be because you didn't originally have that antigen. So the best example I can think of there is the antigens that are on the outside of our red blood cells. Okay, so blood type antigens. Okay, so if you're type O, you don't have any of those antigens on the outside of your blood cell. If you're A or B, then you've got the A antigens or the B antigens. So there's some variability within our species, in other words. Let's see, autoantigens. Okay, so these are usually proteins that are derived from normal cellular processes. Okay, so under normal circumstances, they should not be eliciting an immune response. Okay, so autoantigens, self-antigens. Okay, so normally not causing immune response. But due to some maybe genetic factor or environmental influence, now they are acting to cause an immune response. So things like rheumatoid arthritis causes this painful gnarling of the joints. Okay, that's because we've lost our normal immune tolerance, and now we have aspects of our immune system that are targeting ourselves. Okay. And one other type of antigen that I want to mention that your book doesn't mention are superantigens. And I like to mention them here because we're going to talk about it a couple of infections later. There. I should have answered him probably. There are superantigens. So there are a couple of infections that we're going to talk about later that are caused by superantigens. These are antigens that cause a hyperimmune response. Okay, so this next figure is helpful with that. We're getting ahead of ourselves a little bit into what we're going to be talking about next time. But so normally the antigen here is this black right here. Okay, so sometimes antigen has to be presented. It's usually presented by another cell. So let's say this is a macrophage, for example. For T cells to recognize the antigen, it has to be presented with a certain kind of molecule called an MHC glycoprotein. Okay, and we'll get to that much more soon, so don't worry about that detail. But in a typical, antigen, T cell recognizing an antigen, typically the body would only have maybe a very small number, maybe only a few T cells with a T cell receptor that would recognize that antigen. That's kind of a classical T cell activation. If it's a superantigen, then as many as like 20% of the body's T cell recognize that antigen. Okay, so it's not specific enough. Lots of T cells recognize it. The problem is that causes just unbridled inflammation. Okay, so the cytokine production there from all those T cells is just a lot. Okay, so an infection, the one that I mentioned here, toxic shock syndrome. Okay, shock, when our body goes into shock, that's too much inflammation. The blood vessels get too leaky. Blood pressure goes way down. Okay, it can be fatal. All right, so that's usually a bad situation when you have an infection that's caused by a superantigen, potentially life-threatening. All right, so here's your book's little sum up of the different kinds of antigens. Again, an exogenous antigen could be here a bacteria, maybe it's the proteins on the flagella or something on the outside of bacteria. It could be a virus that's, again, outside of the cell. Okay, those are usually going to be attacked either by antibodies or by some phagocyte. An endogenous antigen is one, here's a virus inside of the cell. Our cells have mechanisms to advertise when they have been infected by a virus. So that's what you see, these little studs lining the cell. We're going to learn more about those. Those are these MHC class 1 molecules that are presenting pieces of protein that's inside of the cell. Okay, so that's how a T cell can come along and recognize that that cell is infected with a virus. And then again, autoantigens, you know, normal cell antigens on the outside of the cell, normally that's not going to be noticed by our immune system. It'll be ignored. Again, only when something goes wrong, then is that recognized. All right, questions? All right, so now we have some groundwork laid as to terminology and where immune reactions are taking place. We'll look at the specific function of B cells. All right, and the B cell receptor. Okay, so remember where we're finding B cells, some in the bloodstream, but primarily in the secondary lymphoid tissue. Okay, so that's lymph nodes, spleen, MALT, mucosal associated lymphoid tissue. Okay, some in the blood as well. Okay, so this is a graphic of the BCR, that's the B cell receptor. Okay, so here's the B cell. And you can see how the B cell receptor, it's anchored into our cell membrane. And then there's what's called heavy chains. So the red portion here, these are the heavy chains. That just means they're higher molecular weight. And then there's a couple of light chains, light molecular weight. You have kind of a constant region down here, more like the stem region. And then on the tips here is considered the variable region of the B cell receptor. So that's the part that's going to recognize epitopes of some antigen. So maybe here's some bacterial cell, for example. Okay, so B cells can directly recognize antigen. And the pictures can be misleading. We always draw one B cell receptor on a B cell. Keep in mind that there's a lot more than that. There's more than 500,000 B cell receptors per B cell. Okay, so it's a crowded place on the outside of the cell there. All of those receptors have the same specificity. So when I first learned that, I thought, well, it seems like it would make more sense for one B cell to have B cell receptors that recognized all different kinds of things. And then it wouldn't take so long to find the right B cell to recognize the right antigen. But it turns out that lots of B cell receptors have to recognize that antigen for the signal to be strong enough to start to cause something to happen. All right, so just one B cell receptor recognizing that antigen, probably not going to be a strong enough signal. Lots of them have to do it. So you want all of those B cell receptors recognizing the same thing. Okay. So B cell receptors, a good analogy maybe is kind of like they're like a snowflake. All right, so you've heard heard it said, oh, no two snowflakes are alike. All right, an amazing repertoire of B cell receptors. You have a B cell receptor to recognize almost any antigen that you could ever encounter. Okay, millions of them. Okay. All right, so what do B cells actually do when they recognize some antigen? Let me come back to how they're activated. When they are activated, what do they do? Well, they get a little bit bigger and they start to divide and they differentiate into plasma cells. All right, and plasma cells are the ones that secrete antibodies. Okay. So I've got a picture to show you of that in just a second. Some of those B cells would differentiate into memory cells. It kind of depends how it's activated, actually. So we'll get back to that. But again, that's a really important hallmark of our adaptive immunity, that you get some memory, so it's much quicker to activate the next time. All right, so let me come back to this in a second. Here's a picture of a plasma cell. I think this is from a guinea pig. This was from your textbook. You know, when you're learning about the parts of a cell, you know, they put like one lysosome and one Golgi and one mitochondria. That's not the way cells really look, though. It depends on what kind of cell it is. So you can tell by looking at this cell that it's specialized to make proteins that are secreted outside of the cell. Because of course, here's a nucleus, you know, big brown thing in the middle here. Most of the rest of the cell here is made of rough ER. Okay, so ribosomes studded on the endoplasmic reticulum, those ribosomes are making proteins that get transported outside of the cell. There's some Golgi in there. That's part of that's function, is to kind of package things and transport it. I think these other blue blobs are mitochondria. It takes a lot of energy to pump out, make so many proteins. A plasma cell that's making proteins is going to be producing about 2,000 antibodies per second. Astonishing amount. All right, so really metabolically active, takes a lot of energy, pumping out antibodies, fairly short-lived. You have short-lived plasma cells that quickly make antibody, and then we have kind of longer-lived plasma cells that do that a little bit more slowly, but they are churning it out. Okay, so we're going to come back to how B cells are activated. I'm just going to lay the groundwork here. But B cells can be activated to differentiate into plasma cells two ways. Okay, sometimes that is dependent on T helper cells. All right, so that's why I don't want to talk about it yet, because we haven't talked about T cells yet. We're kind of mostly talking about B cells to start out with here. And that is probably the most common way. It is the most common way that B cells are activated. Okay, but sometimes B cells can be activated independent of T helper cells. Okay, so in other words, they don't need T helper cells to become activated. When they recognize the antigen, then they just differentiate into plasma cells and start making antibodies. All right, so we're going to look at both of those next time. Okay, so stay tuned for that. But I want to talk about now what's the structure of an antibody and kind of how they work. Okay, before we get to T cells. So if you look at the structure of an antibody, oh, that should look pretty familiar because we just looked at a B cell receptor. Okay, looks the same as a B cell receptor, but antibodies then are not bound to a B cell. Okay, they circulate in the blood. Okay, so antibodies, they're a class of immunoglobulins. Okay, you might have heard that term, immunoglobulin. Basically, antibodies are immunoglobulins. And again, so if you look at the structure, remember, just like B cell receptor, they've got the heavy chain, the inside fragments there, light chain, lighter, lower molecular weight. And then here's some terms that you'll want to be familiar with. The arm region that contains the variable region at the tips. We call that the Fab region. Okay, so that stands for fragment of antibody binding. Right, so that's the Fab region of the antibody. And then the part that's more constant, the stem region down here, we call that the FC region. And that stands for fragment that's crystallizable, which is not as intuitive like how that's important. And there's a hinge region in between those. So kind of like we bend at the waist, you know, the arms of the antibody can bend somewhat to kind of attach to whatever they need to attach to. Okay, there are 5 different, what we call isotypes of an antibody in humans. Okay, other animals maybe have different isotypes, but in humans, there's IgM, so immunoglobulin that comes from the mu region. They've given a Greek letter to the different regions of the gene from where the stem region came from. So IgM, IgG, IgA, IgE, and IgD. Okay, so those are the 5 different isotypes. Okay, and I forgot to mention, this is the picture here on the bottom here. This is a, what do you call this? I'm blanking, space filling model. Okay, so basically every atom is represented here by a little ball. Okay, and the space filling model kind of hopefully helps you understand how this variable region, how specific it can be. Kind of like a really specific puzzle piece. You can see, you know, the shape here that it's going to recognize has to fit like just exactly with some kind of epitope on an antigen. So remember, antibodies very, very specific. Okay. All right. So what do antibodies actually do? It's 5 different functions that we'll describe. Notice that antibodies aren't really killing a pathogen by itself. It's going to do things that are going to bring about the destruction of the pathogen, hopefully. All right, so there's 5 ways we describe this. The first one is neutralization. So this is not drawn to scale because antibodies are proteins and they wouldn't be this big compared to a bacterial cell, for example. But you know, for the sake of illustration, antibodies can bind to some kind of adhesion protein on the surface of some pathogen. Okay, so that could be a spike protein on a virus. It could be binding to a toxin. Could be binding to the outside of bacteria. And when antibodies bind to those adhesion proteins, then that pathogen can't bind to the right cell type that it needs to in order to start its process of infection. Okay, so by neutralization, we'll say it blocks adhesion molecules on the cell or virus surface. So neutralizes them. It makes them so they're not able to start an infection or start doing damage to the host. All right, we talked about this last time. We talked about complement. We know that one of the ways of activating that complement cascade involves an antibody. Okay, so just as a reminder, here's the antibodies here that are binding to some antigen on the outside of a bacterial cell, or some epitope, maybe we should say. And that first complement protein, C1, has a receptor for that FC, the stem portion of the antibody. Okay, and so when it binds to that antigen-antibody complex, that starts the complement cascade. And this is just an overview there, but remember we get bits of those proteins that act as opsonins, bits that act to increase inflammation, and can end up with a membrane attack complex that can poke holes in gram-negative cells. Okay, so that was, we called the classical activation pathway of complement. And related to that, I could have put this, you know, on the last slide there, opsonization, because of course, if complement is activated, some of those bits act as opsonins. Remember what that means? That means that opsonization is anything that stimulates phagocytosis. Or we could say increases phagocytosis. All right, so here you can see the antibodies bound to the outside of some pathogen. There are FC receptors. So remember, that's a receptor for the stem portion of the antibody there. So that macrophage or whatever phagocytic cell can now grab hold of the antibodies so that it can more easily grab a hold of that pathogen and phagocytize it. Okay, it's kind of like a handle. Okay, you can grab a hold of a handle. You know, if that bacteria had a big thick capsule around it, for example, it'd be very difficult for the phagocyte to engulf it. But when there's antibodies caught on there, now it's going to make phagocytosis a lot more efficient. Okay. Something that's, oh, we got these on the same slide here. Something that's that agglutinates, that means it clumps together. Okay, and clumps of antigen are going to be more easily noticed. Okay, they very easily, much more easily get filtered out either in the lymph nodes or in the spleen. Okay, I'll write that. Filtered out. Lymph nodes or spleen. So here, it's more obvious again why it's important that each one of those arms has the same specificity. Okay, if those recognized different pathogens, you wouldn't be able to get them to clump together. Okay, so again, big clump, easily noticed. And then lastly, this involves, this method, the way antibodies work, involves natural killer cells. And it's called antibody-dependent cellular cytotoxicity. We could say facilitated by NK cells. And that's a lymphocyte that I said we weren't really going to mention very much. But essentially that NK lymphocyte has, again, FC receptor protein, so it can grab a hold of the stem region of an antibody that's bound to, say, some bacterial cell. That connection made stimulates the NK cell to release cytokines that brings about the death of that bacterial cell. Okay, so two cytokines called perforin and granzyme. We're going to talk about those cytokines again later because that's the same mechanism that cytotoxic T cells use to kill infected host cells. Okay, so again, and yeah, that brings about the death of the cell. So 5 mechanisms. There may be others that are described a little bit differently, but those are kind of the basics of what antibodies actually do. Okay, if we look a little bit more closely at the structure of an antibody, remember we said that there's 5 different classes, or we could call those classes isotypes of antibodies. And again, we could also call them immunoglobulins. I listed IgM antibody first because kind of the main thing to remember about IgM. It is almost always the first isotype produced in the course of an infection, or we'll say in the course of the immune response. Okay, so once those B cells get activated and differentiate into plasma cells, the plasma cells start pumping out IgM, IgM antibodies. And this isotype looks different because it's a pentamer in circulation in the bloodstream. So 5 monomers make up this pentamer IgM. How do they form? How do they come together? There's a protein called a J chain. So that's this little bridge right here. That's the J chain. These other bonds here are just these disulfide bridges between the monomers. But we think it's the J chain, that protein is necessary for, again, those monomers to come together. We call it an accessory molecule. It's a protein. IgM, and as we'll see, IgA that forms a dimer, both have J chains. So that J chain has something to do with bringing those together. Okay, because there are 10 different places where antigen is recognized, 10 different variable regions, this isotype is really good at clumping pathogen together. Okay, it's really good at agglutination. I mean, you only need 1 antibody to get a big clump together. Of course, you usually have more than that, but also functions in other areas like complement activation, neutralization. It's a fairly small amount of the total antibody in the blood. So really only about 5 to 10%. Typically, if the infection proceeds, then the B cells will switch to making another kind of antibody that's best suited to fight whatever infection it is. All right, so we call that process class switching. Okay, that just means, again, those plasma cells get cytokine signals that causes them to change the kind of antibody that they make. And most often they change to IgG. It depends what kind of pathogen it's fighting, but mostly it changes to IgG because, as you can see, this is the most prevalent kind of antibody in the bloodstream. So about 80% of antibody in circulation is IgG. Okay, so IgM is the first one, kind of the most famous thing about IgM, and it's a pentamer. IgG, most common. And the other interesting thing about IgG is that it's the only antibody that can cross the placenta. Okay, so it makes sense that it's also the longest acting, because we want the mother to pass on some of her antibodies to her baby, and we want those antibodies to keep working in the first couple months after the baby's born, because the baby has a very immature immune system, not as good immune response to a lot of different pathogens. So it's important for the mom to pass some of those along. The only caveat is, if you've heard of Rh disease, sometimes antibodies crossing the placenta is not a good thing. And it's probably not really the concern of this class, but it's sort of interesting how that can happen. Everybody has on their blood type, they're either positive or negative for this rhesus antigen. So I'm blood type A positive, so I have that rhesus antigen. If the mother does not have the antigen and the baby does, because they got it from the father, then the mother can start making antibodies against the baby's blood. Okay, so that can cause the red blood cells to not live as long, it can cause anemia in the baby, it can also cause the death of the baby. So especially after the first pregnancy, when the baby's positive and the mother's negative. So luckily now we have RhoGAM shots that can cause us to not notice those rhesus antigens. And with good prenatal care, we don't have to worry about that anymore. But that's just a little bit of a side. So IgG is good at all kinds of antibody functions. Okay, that's enough for today. Again, bring your thinking caps next time. We're going to get into T cell and B cell activation. Have a good day.